Retrovirus were discovered by David Baltimore and Howard Temin while studying of Rocus Sarcoma Virus, RSV, that cause tumors. RSV is named after Peyton Rouse. In 1911, he found that this virus causes tumors in chickens. Rouse passed an extract from tumor cells over filters that excluded any material larger than a virus. This filtrate produced tumors when injected into healthy chickens. The implications were that viruses cause tumors. But no one knew how the viruses worked to cause cancer. In the 1970s, David Baltimore and Howard Temin proposed that, during infection, tumor viruses integrate their DNA into the host cell DNA. However, this would prove a problem for RSV, which stores its genetic information as RNA, not DNA. They became interested in how RSV might use its RNA as a template to make DNA. To figure this all out, they stripped the virus of its outer coat and used the inner core in a cell-free system. They added all the nucleotides. Thymine was the radioactive tracer. The viral components in this cell-free system produced radioactive DNA. They added RNase, as they knew the radioactive product was DNA because it was not affected by RNase. But the product was destroyed by the enzyme DNase. They incubated the viral cores with trypsin, an enzyme that digests protein. Then added nucleotides to the trypsin-digested viral cores. They did not get any DNA product. Similarly, if they added RNase at the beginning of the reaction, the added nucleotides, there was also no DNA product. They concluded that RSV has a special enzyme that uses RNA as a template to make DNA. It was initially named the enzyme RNA-dependent DNA polymerase, but it later became known as reverse transcriptase. David Baltimore showed that retroviruses have a gene for reverse transcriptase called POL and also carry the reverse transcriptase itself into cells. In his experiment, Baltimore showed that adding trypsin to the viral particles destroys the reverse transcriptase and no DNA is made. The viral particle didn't make any reverse transcriptase as there was no tRNAs, ribosomes, and no amino acids.